Respiration is the process by which hexose sugars such as glucose are broken down by a series of catabolic and redox reactions to form energy in the form of ATP in plants, animals, and some other microorganisms. This is the overall reaction of respiration that you have seen in your previous years of education. However, in this video we will explore this concept a little bit further by looking at its underlying biological reactions. We will look at the separate stages of respiration, and after that we'll summarize this lecture with fill the diagram and past paper questions from International Pearson Edexcel specification. But before that we need to understand the concept of energy inside our cells. Generally, all the energy that is trapped inside of the chemical bonds in organic molecules comes from the sun. The process of storing this energy inside of the organic molecules is called photosynthesis, which forms biomass. Biomass consists of organic molecules such as sugars, fats, amino acids, or nucleic acids. Those organic molecules with energy stored within them are primarily stored in plants, and by the process of feeding, the organic matter, and therefore energy, is transferred to the next trophic levels to herbivores, carnivores, omnivores, or decomposers such as microorganisms. Sugars, fats, proteins, and nucleic acids are then converted into glucose, which is subsequently respired by the organism to form ATP. However, you may ask yourself a question: why ATP? Why not getting, for example, energy from breaking bonds of fats or carbohydrates? First of all, some molecules may be charged or too big to be transported around the cells efficiently, which will make the energy transfer between the cells and organs not optimal. And the second problem is that the energy loss will be much higher. Imagine using the whole glucose molecule to catalyze a small reaction that does not really require that much energy. Here is the reaction that shows the life cycle of ATP inside of our cells. As you may see, the ATP is hydrolyzed by ATPase enzyme, which releases energy which is used by the enzymes to catalyze the reaction. On the other hand, we can also see that the same ADP and inorganic phosphate in a condensation reaction catalyzed by ATP synthase will form the ATP. However, we see that the energy is required for that reaction. And the real question today is, how do we catalyze this reaction? Where do we get this energy from? What are the underlying reactions that will lead to the formation of ATP in our cells? Before we get into the analysis of the respiration reactions, we need to talk about the hydrogen acceptors and coenzymes. The last step of the respiration process, electron transport chain, about which we will talk about later on, requires electrons and protons in order to create a chemiosmotic gradient which allows ATP synthase to phosphorylate ADP to form ATP. So in order to transport those protons and electrons, we need some sort of transporter. We will look at the redox reactions such as reduction and oxidation of coenzymes NAD and FAD, which will be used as our transporters of H+ and electrons. When a molecule gains an H+ and an electron, we say that this molecule got reduced, which also means that the molecule we took that H+ and electron from got oxidized. So when we say that the molecule got oxidized, it lost an electron and H+. In this situation, the coenzymes, our transporters, will gain and lose protons and electrons throughout the respiration process. NAD will get reduced to form reduced NAD, and FAD will get reduced to form reduced FAD. However, you should notice that even though I'm writing NADH and FADH2, you really should be using terms reduced NAD and reduced FAD during your exams, as mark scheme does not really accept NAD and FADH2 notation. We also need to talk about the second type of reaction that will take place during respiration: phosphorylation. There are two types of phosphorylation that will take place in respiration, and one that only takes place in photosynthesis, about which we will not be talking about today. First type of phosphorylation we're going to talk about is oxidative phosphorylation, which occurs at the last stage of the respiration, electron transport chain. This process creates an electrochemical gradient of H+ ions, which is used to catalyze the reaction of ADP plus inorganic phosphate to form ATP. Second type of phosphorylation is substrate level, which occurs when phosphate group is transferred from one molecule to another during enzyme catalyzed reaction. Glycolysis is the first step of respiration that occurs in 3 stages in the cytoplasm that takes place both in aerobic and anaerobic conditions. So it will take place either way, if there is or there is limited supply of oxygen. The first stage, substrate level phosphorylation of hexose sugar such as glucose, describes on how each phosphate group from 2 ATP molecules are transferred to the hexose sugar to form hexose sugar bisphosphate with 2 phosphate groups added from each side. After that, hexose bisphosphate splits into 2 triose phosphates. Luckily, you don't really need to know about the specific enzyme which catalyzes this reaction. Next, oxidation of triose phosphate takes place, which means that it loses its H+ and electrons. Know that those H+ and electrons are actually transferred to NAD to form reduced NAD, NADH, and at the end, the pyruvate would be formed. At the same time, we see that 2 ADPs steal 2 phosphate groups from triose phosphate. You may be as confused as I was when I was firstly looking at this step. Where do we get the second phosphate from to form a second ATP? Once more, luckily enough, you don't really need to know, even though it may seem strange first. That's why at A level we don't really call this stage phosphorylation of ADP to ATP, but rather name it after the redox of these reactions, oxidation of triose phosphate. Now we're gonna talk about regulation of glycolysis. If we were to stop the catalysis of the certain reaction during glycolysis, you should understand that we will stop the whole process of respiration. Would it be aerobic or anaerobic? Because imagine if hexose bisphosphate wouldn't be formed. That's the case with phosphofructokinase. Phosphofructokinase catalyzes the reaction of substrate level phosphorylation of hexose sugar, so the first stage of glycolysis. If we were to stop this reaction, no pyruvate would be formed, and therefore no further reaction would occur. If we were to dive deeper into the theory of glycolysis, we would actually understand that there are several stages occurring during substrate level phosphorylation of hexose sugar, but we don't really need to know about them. This process is actually similar to negative feedback loop, because if we were to produce molecules such as ATP, NADH, and citrate, which we're going to talk about later, they will actually allosterically bend to phosphofructokinase and inhibits its action. Now we're going to talk about anaerobic respiration. This type of respiration consists only out of 2 phases. So glycolysis, about which we already have talked about, and fermentation. During fermentation, pyruvate produced in glycolysis is reduced, so it gains an H+ and electron. This leads to the formation of lactate in animals and bacteria. However, in plants and yeasts, our pyruvate is going to be reduced and decarboxylized. So at the end, we're gonna form 2-carbon molecule ethanol and CO2 as the waste product. Later on, our lactate is going to be transported to the liver via blood, where it will get oxidized in order to form pyruvate, or it also can be converted to the glucose. Both of those products will be reused in the glycolysis in order to renew the cycle. This process is also called gluconeogenesis, which occurs, as you know, in the liver and requires a lot of oxygen, which is supplied, for example, after a heavy exercise, when most of your muscle cells were switched to the anaerobic respiration. That's actually the reason for your post-exercise heavy breathing, because your liver requires oxygen to convert this quite acidic lactate into the pyruvate and glucose, so that they can be, again, be used in aerobic respiration. So now, as we finished with glycolysis, we are going to talk about the second step of the respiration, link reaction. It mainly occurs in the mitochondrial matrix. The pyruvate that was formed in the cytoplasm during glycolysis is transferred through the facilitated diffusion down the concentration gradient to the mitochondrial matrix, where it gets decarboxylized. Which means that a molecule lost carbon in the form of CO2 in this specific example. So when our pyruvate loses the CO2, it forms acetate, which is oxidized, so it loses H+ in electrons, which are transferred to the coenzyme NAD to form reduced NAD, NADH. At the same time, coenzyme A, the second type of coenzyme, will bind to the oxidized acetate and will form acetyl coenzyme A, which will be used in the further step to aid the next reaction. After the formation of acetyl coenzyme A, the oxaloacetate is going to accept the 2-carbon molecule from acetyl coenzyme A, so basically acetate unit, to form citrate, 6-carbon molecule. Note that during this reaction, coenzyme A unit is getting reused in link reaction. After the formation of citrate, regeneration of oxaloacetate is going to take place, when this 6-carbon molecules is going to be oxidized by 2 types of coenzymes that we talked about, namely such as NAD and FAD. 3 molecules of NAD are going to steal 3 H+ in electrons from the citrate, and 1 FAD is going to take the 1 H+ in electron to form 3 NADH, so reduced NAD, and 1 reduced FAD. The second thing that will take place is decarboxylation of that intermediate. 2 CO2 molecules are going to be released, and the 6-carbon citrate is going to form 4-carbon intermediate. However, we are not really done yet. The last step is substrate level phosphorylation of ADP to form ATP. Previously, you only have seen a molecule such as glucose getting phosphorylized. However, in this situation, ADP is phosphorylized. You need to take a note that phosphate group is transferred from the 4-carbon intermediate to the ADP. After all of those processes, the new oxaloacetate is going to be formed, which will be ready then to take a new acetate subunit from acetyl coenzyme A. Here is the simplified diagram of first 3 steps of respiration to get you to know the overall production of all reduced coenzymes that will be used in the next step of respiration, electron transport chain. Try and remember the numbers of reduced coenzymes and the products that were produced during all of the steps of the respiration per 1 glucose molecule and per pyruvate, because these are the common questions on respiration that you may find in your exam. So now, since we have counted all of the coenzymes that were present in the first 3 steps of respiration, we need to understand that all of them will be transported to the inner membrane of mitochondria. So the first step of oxidative phosphorylation is that the high energy electrons are harvested from reduced NAD and FAD and are donated to the first protein complex if it is NADH, and to the second protein complex if it is reduced FAD. Then, as those high energy electrons are moved through the electron transport chain, The energy produced by this movement is used to pump H+ from the mitochondrial matrix to the inner membrane space against the concentration gradient. This makes the intermembrane space more acidic and more positively charged than the matrix, thus forming an H+ electrochemical gradient, which is also known as proton motive force or chemiosmotic gradient. Because there is such a high concentration of H+ in the intermembrane space and such low concentration in the mitochondrial matrix, ATP synthase will act as the H+ channel, so that the H+ ions will move from the intermembrane space, so higher concentration, to the lower concentration into the mitochondrial matrix. We also need to add that this movement is passive and also facilitated by the ATP synthase. And this movement of them with their concentration gradient will catalyze the reaction to phosphorylate ADP to form ATP. When our high energy electrons move throughout electron transport chain proteins, they lose their energy state. Therefore, at the last protein of electron transport chain, we would have a low energy electrons. And because they are low energy electrons, they cannot really be reused in the electron transport chain. That's why we give away those low energy electrons on the formation of H2O. This step is crucial because it prevents the backup of electrons in the electron transport chain. If oxygen is not present to accept the electrons, those low energy electrons, the electron transport chain and therefore oxidative phosphorylation would halt. This will occur because the proton gradient cannot be maintained, and because of that, our cell may die or simply shift to the anaerobic respiration. Now we're gonna move on to the fill the diagram question. Signify the number and type of reactants and products of each reaction, as well as the place where the reaction takes place. If you are satisfied with the answers that you gave up in the diagram, congratulations! Now we are going to be subjected with the past paper questions from the previous year from Specification of International A-Level Biology Pearson Edexcel. Please stop the video when the question will pop up because the answers will show up fairly quickly.